Now that we have an overview of the different dosage forms available, let's look at the considerations in designing a dosage form. First, let's get our terminology right. A drug is a substance administered to human or animals for prophylactic, diagnostic, or therapeutic purpose. In the pharmaceutical industry, a drug is also known as an active pharmaceutical ingredient or API. A dosage form is a system that delivers the drug or active ingredient. Besides the drug, the dosage form also contains other substances called excipients which impart desired properties to the dosage form. One drug can have multiple dosage forms. For example, aspirin is commercially available as normal tablets, effervescent tablets, and enteric-coated tablets. The main objective of dosage form design is to achieve a predictable therapeutic response to a drug included in a formulation, which is capable of large-scale manufacture with reproducible product quality. The drug product should be safe and effective, stable, and acceptable to patients. To meet those objectives, the dosage form should be physically and chemically stable, be protected from microbial contamination. The dose of the drug should be uniform so that the patient gets a consistent dose each time they take the drug. The product should be acceptable to both the prescriber and the patient. And the product should have suitable packaging and labeling. When designing a dosage form, many factors have to be taken into consideration. There are biopharmaceutical considerations. This refers to how the body handles the drug. There are the properties of the drug itself. And there are also therapeutic considerations related to the type of disease or patients who will be using the drug. Earlier, we have already looked at the various routes of administration. Some considerations and characteristics of drug absorption through these routes are summarized here. The oral route is often the most convenient route to deliver systemic acting medications. However, there are many factors which can affect the absorption of the drug, and these factors need to be considered when designing a dosage form. The rectal route can be used for both local and systemic effect, but absorption through this route is irregular and less predictable. In emergency cases, or when a drug cannot be given via the oral route, an injectable route would be very useful. The topical route is mostly used for local effect, although there are also transdermal products available. The respiratory route provides a high surface area for drug absorption and is especially useful to deliver medications to the lungs. Different dosage forms have different times to the onset of action as summarized in this table. For example, IV injections work almost immediately, whereas oral dosage forms may take minutes to hours. So an appropriate dosage form is also chosen based on how quickly the drug effect is required and for how long. The properties of the drug itself must also be considered in dosage form design. Many drugs have an unpleasant taste. This is a problem because any drug that is given orally must be acceptable to the patient. There are several ways to overcome the unpleasant taste of a drug. A patient can taste the drug when the drug dissolves in water or saliva and binds to taste receptors on the tongue. So one method is to use water-insoluble derivatives of the drug. For example, chloramphenicol is a bitter-tasting antibiotic, but its derivative, chloramphenicol palmitate, which is insoluble, is tasteless. However, take note that by chemically modifying the drug, it is considered to be a new active pharmaceutical ingredient. Another strategy to mask the taste would be to add compatible flavors to the product by adding flavorings, perfumes, sweetening, and coloring agents. Enclosing the drug in a capsule would also help to prevent the drug from reaching the taste buds. If it is a tablet, it can be sugar-coated. The drug could also be made into an effervescent tablet since the flavorings and carbon dioxide would help to mask the bitter taste. The particle size of the drug is also important. There is a wide range in particle sizes used in dosage forms. The particle size used in parenteral suspensions and aerosols for inhalation are generally smaller. For oral and topical formulations, the particle size can be bigger. Particle size influences dissolution and absorption rate. Smaller particles can dissolve faster and therefore get absorbed faster. Particle size also needs to be uniform to ensure that the drug is distributed uniformly within the product. Particle size also influences stability of the product. Smaller particles tend to agglomerate or clump together and may not flow well. But in the case of suspensions, large particles in suspensions will tend to settle faster. The solubility of a drug is also important. All drugs, whatever route they are administered by, must exhibit at least some aqueous solubility for it to be effective. The solubility of a drug can be improved by using a more soluble salt or ester derivative of the drug. This is considered as a chemical modification of the drug. Micronizing, which is to reduce the size of the drug particles. Complexation of a drug with a water-soluble carrier can also increase solubility. For example, complexation with cyclodextrin. Solid dispersion is to disperse the drug in a water-soluble polymer. So when the polymer dissolves, that drug is also dispersed and can dissolve very easily. Most drugs will need to pass through biological membranes in order to be absorbed and reach its target. For example, orally administered drugs will need to cross the cell membrane in the intestines and the blood vessels. Therefore, it is important for drugs to be permeable through biological membranes. The permeation rate depends on the size of the drug molecule. Small molecules will pass through the membrane more easily. Biological membranes are made of fats or lipids, so drugs need to have a balance of aqueous and lipid solubility in order to cross the membranes. Molecules that are charged would not cross lipid membranes easily. The partition coefficient, or P, measures the relative affinity of a compound for the lipid versus aqueous phase. N-octanol is used as a lipid and water is the aqueous phase. The drug is added and shaken together with the N-octanol and water. Then the concentration of the drug in the N-octanol phase and the concentration in the aqueous phase is measured. Partition coefficient, P, is the concentration of the drug in octanol divided by the concentration of the drug in water. We often convert P to log P. If log P is less than 0, it means that the aqueous phase has a higher concentration of the drug compared to the octanol phase. This means that the drug is too hydrophilic, which means water-loving. It would not penetrate lipid membranes easily. On the other hand, log P should also not be too high. A high log P means that the drug is very lipophilic or lipid-loving and would most likely have very poor solubility in water. Whether a drug is permeable across lipid membranes also depends on whether it is ionized or has a charge. Many drugs are weak acids or weak bases. Depending on the pH, they can be ionized or unionized. The ionized form is more water-soluble, whereas the unionized form is more lipid-soluble. A drug may be a weak acid. Let's represent it as Ha, A for acid. This chemical equation shows that the drug can exist in two forms, A- and Ha. In an acidic environment, the drug exists as Ha and is unionized. You can see that there's no positive or negative charge on the Ha. The unionized form is more lipid soluble. In an alkaline environment, the drug exists as A-. There is a negative charge on the A-, So this is the ionized form, which is more water soluble. On the other hand, some drugs are weak bases. Let's represent the drug as B, B for base. In an acidic environment, the drug exists as BH+. There is a positive charge, so this is the ionized form, which is more water soluble. In an alkaline environment, the drug exists as B and is unionized. This is the more lipid-soluble form. The proportion of ionized to non-ionized molecules is determined by the pH of the solution and the pKa of the drug. pKa is a measure of how acidic or basic a drug is. Many drug substances can exist in more than one form. Amorphous means that the molecules have no distinct arrangement, whereas crystalline means that the molecules are arranged in a crystal structure. The crystal structure could also include water molecules. The anhydrous form does not contain any water molecules, whereas the hydrated forms contain water molecules. These pictures show how a drug can exist in different crystal forms. The chemical structure of the drug is the same as shown here, but the molecules of the drug are arranged differently in the different crystal structures. The ability of a substance to occur in different crystal forms is known as polymorphism. The polymorphs can have different properties, including different solubility, dissolution, stability, powder flow properties, and compressibility. Therefore, it is important to select the correct crystal form. Polymorphic transitions from one form to another may occur during processes that involve heat or moisture. Therefore, it is important to be careful when handling and processing such material. Some drugs may decompose under certain conditions due to sensitivity to heat, light, oxygen, or moisture. Incompatible excipients and improper packaging can also affect drug stability. So, to maintain drug stability, for drugs that are sensitive to moisture, methods of manufacturing must be selected to avoid exposure to moisture. Antioxidants can be added to drugs that are oxygen sensitive. Light-sensitive drugs should be packed in amber or opaque packaging. For pH-sensitive drugs, buffers can be used to maintain the desired pH. Preservatives can be added to prevent microbial growth. And excipients or packaging materials should be selected carefully to prevent adverse interactions. Some drugs may be hygroscopic, meaning they absorb moisture over time. These drugs would require low moisture manufacturing environments. If the drug powder is not able to flow well during manufacturing, flow agents such as fumed silica may be added. The compressibility of the powdered materials is important when manufacturing tablets. Poor compressibility could potentially contribute to tablet defects. Finally, therapeutic considerations related to the type of disease or patients should also be considered in drug formulation. What type of disease or illness is it? Would it require systemic or local delivery of the drug? How long do we need the drug to work for? How quickly must the drug take effect? And is the dosage form appropriate for the patient? For example, children may not be able to swallow tablets. After taking into account all the considerations, the drug company will need to select the most appropriate dosage form, select the method to manufacture the dosage form, select the suitable excipients, which should be compatible with the API, develop the formula, so what proportions of each ingredient should be added, make the dosage form, select suitable packaging materials, and carry out stability studies. Just to give you a peek into the many different types of excipients, some of them are listed in these two slides. We will be covering their uses in the different dosage forms in the upcoming topics.